was a multiple collaboration between actually multiple labs in which they harness these single cell technologies to profile and create a census of all the cellular types in the human cortex. Also, recently we have published about all the cellular types in the mouse brain. So, if we think mathematically about this problem and to really harness all these promises of single cell technologies, there are many challenges that need to be overcome. So let's think mathematically. What we have is certain data that can be cast as a matrix, and each column of this matrix represents one of these cells that we profile. And then the rows are the features that we measure. And I say features because if we profile the transcriptional landscape, these are genes. If we profile the chromatin accessibility, these are the peaks or chromatin regions. And what I want to show in these graphs is that as I have been telling you in the last couple of years, or well, no, last 10 years, we have been pushing the technology higher and higher, and we are able to profile more and more cells in each experiment. But with this increase in the number of cells that we profile, what is going on is that the ability to profile or to observe each individual feature or each gene has decreased. So we have increased the sparsity. We have gained our ability to profile many more cells, many more observations, but at the same time, we have reduced our ability to observe each feature. And this imposes conditions or requirements on the computational algorithms that need to analyze this data. On the one hand, they need to be scalable. They need to be able to process millions of observations. And on the other one, they have to be able to deal with sparsity or reconstruct the underlying biological variability from this sparse data. But this is not just the only, sparsity is not the only problem and scalability. We also, within our measurement, have multiple batches, technical effects. The experiments originate from multiple genotypes. They have different cell types within our experiment. And now I'm pushing you to think that we also have multiple modalities that we are profiling. But if we can achieve this goal and this promise of profiling this or of harnessing single cell experiments, the reward is tremendous. Because what we can, let me show you an example. Let's think on disease or Alzheimer's disease. We know that pathology in Alzheimer's disease accumulates decades before the clinical symptoms are shown. All right, so we could think as an homeostatic cell, like the microglia I was describing, that is evolving as time evolves in us, in humans. But some insult happen in the brain, pathology start accumulating, and these cells become inflamed or in an inflammatory state or a disease response. If we can identify these cells, I'm not pointing there, I was pointing there. If we can identify these cells and create therapeutic cell target interventions years before the clinical symptoms show up, we can start maybe thinking about treating disease. So again, there are challenges to analyze single cell data, but the rewards are tremendous if we can achieve it. Okay, so that was kind of the broad overview of why should we care and what we care about all the work that we did. So what I'm going to show during this talk, and this is the core of the talk, is I'm going to showcase some machine learning algorithms that we developed for the analysis of single cell and multimodalities technologies. I will try to be pedagogical, actually. And then at the end, I will showcase its application to the study of Alzheimer's disease. So there is a lot of material I like to cover. So let's see how it goes. All right. So again, we need to think about this data as originating from a matrix in which we profile cells and multiple features. And each of the cells is associated with certain covariates. We have the batch or the laboratory from where the cell was collected. There are technical factors, and the cell has a cell type. And this is the observed data, but what we would like to actually infer is the latent or hidden state that I'm represented with the variable C that represent the state of the cell and maybe the cell type. Co-learning, while we are inferring this latent state, all the technical factors that are modifying or perturbing the experiment. And the way we are going to do it is taking a probabilistic description. We are going to infer this C, this latent representation, given the observed data. And again, these will represent, these are actually layer 5 IT cortical neurons, and this is actually how the embedding of these cells looks. And given the embedding, given the observed data. So if we achieve a probabilistic description of the data, this probability description will allow us to infer or query our model at many different levels. And I want to portray two, and this is a kind of an organizing principle of our algorithms and of the talk you will see throughout the talk. We can query these probabilistic models at the cell level. And at the cell level, the queries will be low dimensional. We would like to infer the embedding or the manifold in which the data lives. We would like to harmonize or correct that the data originates from multiple laboratories, and the embedding should just capture biological variability. And if we have labels, we would like to take a new data set and maybe do label transfer. Those are some of the queries that we can do at the cell level or low dimension. At the feature level or high dimensional, we would like to denoise the data. All of these features, that's why it's high dimensional. We have 36,000 of genes that we profile. We would like to get the average expression levels, maybe within a cell type. We would like to get the actual expression level in the cell if we remove the technical artifacts. And if there are missing values, we would like to impute them. So these two levels of analysis is like kind of the what I call the organizing principle or the achievements that we would like to make from our algorithm. And one of the most successful, I like to think, because that's what we do, algorithms that is able to do this is scVI, which is an unsupervised way of learning a representation of single cells. And scVI takes us as an input a matrix that is genes by cells and infers the low dimensional representation, removing all unwanted dimensions of variation. And then reconstruct it or denoising the data, allowing you to compute differential expression and many different things. So I'm going to show you next in a couple of slides, like these simple, like pedagogical slides of the math behind scVI, trying to build from what was the first early algorithm that shows scalability and a machine learning algorithm up to our multimodal algorithms that we published last year. And for that, I'm going to take a simple example of probabilistic PCA. I'm going to show you a little bit the description, the framework, the probabilistic framework that we use, how we do parameter inference, and then finally ending with, scVI is based on variational autoencoders. So let's walk. So we care about the probabilistic description of the data given, and the object that captures all the information given our probabilistic description of the data is what it's called the joint distribution. So one way to represent a model, for example, I'm showing you here on the left, is to say there is a latent variable C, that this is what I have been showing you all along the top, one for each cell. And then there is an observed variable X. X is gray because it's observed, and then C, within this circle is white because it will be inferred or it's a latent variable. And the arrow represents conditional dependencies between the variables. Then this plate diagram or this square with the letter N means that these variables are sampled identically independently distributed N times. Okay, so there are n cells, each of them with its own observation, and we will have a latent variable C, or the embedding, for each of them. One method that creates a linear representation, this is our, the most math-loaded slides, just to get a sense of, like, cater to all the audiences, from the experimental biologists to the computational one. The conditional dependence that I was talking about here is reflected in the factorization of the joint. We have a variable x that depends on C, and then we have certain probability distribution for C. And if we consider the following model, C samples from a normal distribution, this is random, and then we transform linearly C by many tricks, we multiply linearly, and then we corrupt it with noise. This is the actual representation of probabilistic PCA, which is a way of representing linearly our data in a probabilistic framework and saying that it's corrupted with noise. How are we going to do, given our model? This is what I just told you, actually, is called the generative model of the data. How do we generate data? Like, we sample from C, from a normal, we get some random noise, we multiply by a matrix, we sample with other noise, we get data. But how do we revert the model, and how do we infer the parameters of this model? So we are going to follow a Bayesian approach. And our parameters will be the variance of the observed data, the matrix that we are multiplying, and this latent structure. So taking a Bayesian approach imposes that we give to each of these parameters a prior distribution. Okay, and this is more like just giving a prior. So our prior will encode our prior belief on the parameter. Let's say the variance is positive. So the distributions that we have, the prior will have to have positive support. And given, if we have data from the Allen Institute, and I have analyzed it and it has no batch effect, there's almost no noise. So you will want sigma to be small, so you will adjust your prior. But this probabilistic description, as I'm saying, is statistical in nature. So what we get by doing Bayesian inference is that we not only are going to infer the parameters, but we are going to infer the distribution or the confidence interval. So by taking a full probabilistic model, we are able to represent our parameters statistically and infer its distribution. And what I'm showing you at the bottom is now with the prior and the way the likelihood or the data is represented, we have the joint distribution that I was telling you capture all the information in the probabilistic model. Okay, so how do we infer these parameters of the model? We use Bayes' rule to compute the posterior distribution given the data. Or the tail would like to say, like, how many are statistician and how many, this could be for some of you like a simple review, but the tail would like to say is like, we have a prior on the parameters that encode our belief, and we update the prior with the likelihood of the data, and that's what are the parameters given the observed data. Also, the tail will say is that computing this integral or this quantity here is impossible in many cases, it's intractable. So we have to resort to approximate Bayesian inference method to infer the parameter. So again, we care about inferring the parameters, we care about finding the posterior of the parameter, the distribution of the parameter given the data, and we typically say this is proportional to what I have been telling you, the joint distribution. Okay, but again, I have been telling you this is difficult, we cannot do it. So how do we do it? We are going to resort to an approximated Bayesian inference method called variational inference. And what variational inference does is the following. We are going to propose an approximation to the posterior. All right, to this distribution of the parameter, we are going to propose an approximation. And then we are going to minimize the distance between our approximation and the true posterior. And we are going to minimize the distance at measure that we're going to use is the KL divergence. All right, so we have our posterior distribution and our proposal, variational proposal, we minimize the distance, we find the distribution of our parameters. But the thing that I have been telling you is that we never know the posterior distribution of the parameters given the data, because there was this quantity that was intractable and we were not able to compute it analytically. So what do? I'm telling you, okay, do this, but minimizing the distance from the proposal to the actual posterior of the parameters is equivalent, it can be shown, to maximizing what is called the elbow. And this quantity doesn't involve that intractable term that we couldn't compute, okay? So, and the elbow is kind of a balance between two terms. One is like the likelihood, and we want to maximize the likelihood, we want to maximize the fit from the data, but at the same time, we want our posterior to be close to the prior. So again, KL divergence is a divergence that measures distances in probability distribution. So by balancing these two terms, we are able to fit or find the distribution of our parameters, the posterior distribution. Okay. Yes.
What if the true posterior is not in that family?
You find an approximation to, so it could be that.
Can you repeat the question?
What happens if the
true posterior is not in the variational family?
So let's say that this is the true posterior, or the space. This is the space of distributions that are concordant with the factorization that I've shown you. And the true posterior lies here. Or no. Let's say the opposite. This is the space of Qs. All the possible distribution given certain parameters that you can optimize or change. And let's say this is your true posterior. What you just said is that the posterior is outside the family that you can represent. So what you find is that the best distribution close to the posterior, but clearly you cannot find you find an approximation. Like that's why it's called, that term was called the variational proposal. It's a proposal that you will try to make it close to the true posterior.
So you just minimize the K between that.
No, you always minimize the KL between, which is maximizing the elbow.
Yeah.
But what you find is the closest KL, let's say divergent, but it's not. Let's say if you had a more flexible distribution, like in your proposal, and the space is like this, okay, you will find the true posterior. Okay. But you find the closest in this like multidimensional functional space.
I mean, you could learn that area right there, but yeah, I guess we're good. Not good.
Sometimes for mathematical simplicity, you propose even variational proposals that don't have the right factorization. Oh. But you do it because you can compute it analytically. It's easy. That was called mean field variational impact.
Okay.
And that carried on the field for a while. But you knew that even if the factorization is incorrect, your true posterior will not be, but you will be close enough. So what I'm showing you, and I have a lot of material, so let me go. Okay, so MNIST, this is the most machine learning like data set you can have, so it's all the digits in a square drawn handwritten. So now our cells are each digit handwritten. So we can use this probabilistic PCA and infer the posterior of the parameters and compute for each digit the variable C, which is embedding, and see that, okay, all the here with colors is each of the digits. Some of the digits are separated, but some of them are mixed. So it's computing some representation of the data up to a certain point. And that's not due to the variational approximation, it's due to the expressivity or the capabilities of a linear model. There was something that came in 2014, and it took actually Romain 4 years to figure it out and get it published, which was called a variational autoencoder. A variational autoencoder is a more flexible, more than linear probability model, in which you have, again, a latent variable C, and then you are going to transform with a flexible function, which is a neural network, this C to represent the mean of the observation. Okay, so then you have C, low dimensional. Through a neural network, you reconstruct the data. And the variational proposal, you also want it to be flexible and encompass more possible candidates, posterior distributions. So you are going to take the data X and you are going to transform it to obtain a mean and a variance of our posterior parameters through neural networks. So this idea of adding neural networks into the problem allows you to be able to represent nonlinear interactions and be more flexible and have a model that have better capacity in inference. One of the things that it also gains us is that for each of the observations, for each of the cells, to compute the embedding, we have just one neural network. And this was called that you were amortizing inference. Why is that? Because before, you could have as many as n parameters. But now you have as many parameters as the neural network has. Once that you fit these parameters, you can fit all the observed data. Okay, this idea of amortizing inference, just having a fixed set of parameters, was something else that was really amenable of this type of model.
So is it that by using a neural network there, you're basically not defining a specific distribution, like, right? Like you can have a power law distribution, or you can have a normal distribution. So you're basically not defining a priori what's the shape of that, but you're rather letting, the neural net figure that out and how many parameters you need.
Yeah, in certain, you're finding the transformation, the mapping, a more flexible mapping. So, when this was presented early in new RIFs and the implementation, one of the simple cases will show you a mixture of Gaussian that you could represent, Gaussians. And then after the neural network, you will corrupt it again with noise, but you can get shapes like this. So you will be able to represent the data with a simple underlying representation and be able to flexible learn it. While probably if you learn this with PCA, you will get like one Gaussian here, one here. So you will learn something, but it's a linear transformation, which is an affine transformation of a norm. What happened with that? Okay, all right. So just what is called autoencoder, because there was this neural network framework in which you have the X, which is the observed data. You condense it to a low dimensional representation, and then you reconstruct it like it to X. So this ability of this idea of fitting X to its reconstruct version was called autoencoder. This is called variational autoencoder because it arrives naturally from a probabilistic description. In the autoencoder, you didn't have nu or sigma, like you didn't infer quantities, statistical or probabilistic quantities in them. Okay, so if you fit, a variational autoencoder to MNIST, well, now the digits are more separated. I hope that you can see and you believe me that now we represent the data more flexible. It's not perfect, but it was better than before. Okay, so going back, SCBI was actually the first variational autoencoder-based framework to analyze single cell data, in which we will take a cell by gene matrix, we will obtain the variational posterior, which is the embedding that I was showing you before. This variational posterior of the parameter C will be what captures the latent representation of the data, to then reconstruct the data in a denoise manner. But one of the cool ways of SCBI was that we really, at that time, Romain really modeled the data. What is the data that is coming from the sequencer? It's like multivariate count data represented by a Poisson distribution. Okay, so that multivariate data is multivariate count data is embedded into the distributions of the model. Then we have certain parameters, certain distributions and parameters within the model that captures the sparsity. What is the likelihood of observe a gene or not? We have the way to infer the latent representation. We correct for library size, or we infer a parameter that represents library size, and the transformation between the latent state and the mean of the counts is through a neural network with this flexible representation. Okay? So scBI was the first one of many instances of probabilistic algorithms enclosed within the scBI tools frame. And that's when I joined actually the Joseph lab, when scBI tools was being cast and created. And scBI tools is the framework, because what a framework should do is to abstract something from the user. What scBI tools is doing is abstracting variational inference. It gives you like a set of primitives to build your own models, but then you could carry on and use like the inherent machinery of scBI to do variational inference. Together with many more things like differential expression, then people start building up. Now that there are like 15, 20 models out there, like can do processing of spatial transcriptomic, processing of ATAC, processing of many different things, together with processing of multimodal data set. And that's where scBI tools was not the core of my Romain did scBI. Adam Gayoso focused really on building all the infrastructure of scBI tools. And I would care with a group of people, TAG in particular, to do multimodal algorithm, extending scBI to process protein information, protein expression, chromatin information, ATAC-seq, and gene expression data. And the idea that scBI wanted to capture was that now we have all of these multiple views, if you want to think like in machine learning terms, views of the data, how to create a notion of cellular identity that encompasses all of them. And at the same time that scBI was being created, there was this multi-omic assays that now they are commonplace, in which you profile jointly in each cell 2 modalities. But then you have some assays in which you profile 2 modalities. Then we have a lot of transcriptomic data. You have some ATAC. So scBI is able, MultiVI is able to take jointly profile or singly profile data. You throw it all in, and we infer the latent representation from all the data. Okay, so let's walk a little bit through some of the nuances of MultiVI. And if I say I'm saying scBI, but now I'm talking about multimodal. Okay, when we think about transcriptional data, gene by cells by gene matrix, there are certain intricacies of the data. One is the sparsity. You miss many of the observations, and then you represent the observations as counts. If you do a CITE-seq experiment, you collect transcriptional and protein expression data. So now protein has a different set of challenges. One of the challenges of measuring protein is that you have a non-zero background. So to really identify the signal, you need to fit a mixed model of 2 components to really, for each protein, identify background and signal. Then, if you think about chromatin, I don't even have a diagram there because we know chromatin data is even sparser. Like for a particular loci that the TM5 transposase is binding, you could have 0 or 1. When you go to a peak, which is a finite region, okay, you could have many more observations, but intrinsically, it's more sparse. So MultiVI tries to account for all the nuances in the data and really model it, while again, capturing this organizing principle, a low dimensional representation that can be queried, and a high dimensional representation that will allow us now to denoise genes, protein expression, chromatin, compute differential expression, differential accessibility all at once. In statistical terms, now that we know more and more, the low dimensional representations, let's compute the posterior probability of the cell, given the data and maybe batch or covariates. The high dimensional representation that we compute with variational autoencoder is let's compute the probability of the data, the profile of the gene expression, given the data and the batches, the covariance. So let's compute the posterior, or in this case will be the predictive distribution. How do we do it? This is the layman's audience, like a slide in which for each of the data types that we process in MultiVI, we compute a latent embedding of the data. And then we try to merge it into a joint representation. This joint representation, again, remove all unwanted directions of variation, like batch or technical effects. And again, having this high and low dimensional overarching principle. And we do it by, again, finding, proposing a latent space with a multivariate distribution. This was not novel. But for example, when we analyze chromatin and transcriptional data, we have for the transcriptional data a negative binomial. We have a set of counts that we want to model. For the chromatin accessibility, we model it with the Bernoulli. Is there reads or is there accessibility at one particular region, yes or no per cell? Again, from the latent representation to this mean expression of each of the observation in each cell, we use neural networks to non-linearly transform the data, the latent space to the data. We have feature-specific effects, like, and we also infer library size. We co-learn all together. And the feature-specific effects, for example, could represent if there are two regions in the genome and they have different GC content, they could have inherent ability for the TM5 to bind there, or inherent mean accessibility without even the cell type variability. So give me a couple more slides, one slide more. Okay, and just to tell you the last detail of MultiVI is like I told you that we create for each representation or each data type and each observation, we create its own latent embedding. Okay, and then we bring it together. We do it by computing the mean of the individual embeddings. This is the joint representation, and at the same time, penalizing the distance between the two representations. So we want them to come close, and then to compute this joint consensus representation, we compute the average. So what comes next is a little bit of performance. So you had a question before.
I'm trying. So if I got this right, for the individual modalities, you give it the feature set for the feature space for that particular feature set. For example, chromatins may be modeled by one distribution, and then you have transcriptional variation by another distribution. Assuming that these distributions, I'm wondering how generalizable is this for things if you're not interested in chromatin, if you're interested in
different omics.
Yes.
Well,
I'm trying. So if the distribution is the same as maybe chromatin is, let's say. Well, let's say.
Yeah. So you're going into some of the okay. You will have to give me more time. But some of the prerogatives, let's say, that we have on the inference was that think on the inference, you start with random parameters, then you converge. So you converge to a point in which all these distributions are equal. Yeah. That's the idea that we have. Right. Even we compute the average. If you compute the average between two equal things, you get the same. So in equilibrium, if you achieve that, you are good. And some of the things that we balance, and there was a paper I cannot recall now that showed this, is that if one of the modalities is more informative than the other one, let's say chromatin has some information for some cell types that RNA doesn't, to be able to reconstruct the data, you carry that separation all over through all the distributions. So if there is one of them that it's slightly more informative, you will kind of share information across modalities. This is not always the case because now I have fit this to whatever, and in cases in which one modality is, let's say, in mutual information terms, but it doesn't matter, like really informative, and another one is poorly informative, you don't mix them well. Like if there are like But that doesn't happen typically, like in chromatin, RNA, protein expression with, let's say, 100 markers. Like they all carry enough information to be able to glue them together and share this information.
But when you're calculating the mean, how do you give that a chance? So let's assume that the RNA has more importance than the attack.
Because then you need to reconstruct the data. So you have a term. Let's say you have a blob in RNA. Let's say the RNA is like this. Okay, and the chromatin is like this. Okay, if when you join them, you get this representation, you will lose information here. So your reconstruction term will be penalized. However, if you end up fitting here, you are not penalizing, like the neural network of the decoder, the one just learn, okay, independently of this, reconstruct something that is homogeneous, more or less. But in the other one, you really need it, because that shape is carrying on, some variation, different gene expression programs, etc. Okay. So quickly, does it work? Well, here, what we did, we took PBMC's datasets from multiple laboratories for multiple technologies, multiple RNA technologies, 10x chromium, 10x B3, B2, different depths, drop-seq, seq-well, like multiple replicates, ATAC plus RNA, RNA and ATAC, and different cell types. And it does a pretty reasonable job, actually, I would claim, integrating and removing all these sources of variation. And this is not, this comes from multiple labs. So if you don't do anything, the data has batch effects and doesn't mix. So it's truly trying to reconstruct it. Then just because we can do it, we reach out to the Allen Institute for Immunology. They have this assay called TA-seq in PBMC cells also that profile RNA, ATAC, and protein in the same cell. But if you have all the information, then you can tease it apart and create data sets that just have ATAC, just protein, ATAC protein, okay, all the possible combinations. And then you just dump that in the algorithm, you integrate it, and you can see that cell type information is preserved after the integration of all of these multiple data. Although we quantify that, I'm not showing that there are these metrics of how well you integrate, what is the probability of finding multiple batches, etc. Something maybe to talk about quantification that is more exciting to me is the following. We did another experiment with PBMC cells in which for each cell you have, it's a multi-omic experiment, so you have RNA and ATAC profile in the same cell. So you can take some cells and split them, create kind of sister cells. Some cells that just carry the RNA and the ATAC, but you know that this cell is this one, etc. You can run through the algorithm, the pair and the unpaired, artificially unpaired cells, and measure the distance between the sister cells. And we compare with multiple algorithms, and what you could see is that the sister cells are really nearby. And compared to, at the time, multiple Surratt, different algorithms, like adaptations, because at the time they didn't work with individual assays. The only one that was comparable was COBOL, which was also a BAE-based framework. Another experiment that you could do is the following. You can take one particular subpopulation, one cell type, these are B cells, you have to trust me, and remove all the chromatin information. And then compute the probability of the accessibility given the transcriptional information, kind of imputing the missing chromatin information. And then assessing the following. This is a differential expression of B cells, those cells, against all the other PBMCs. And this is the observed difference and the predicted difference. Perfect will be aligned, but this is quite aligned, and it's showing that the most differential accessible peaks are well imputed. In addition, if you compute the kind of the base factor or some type of posterior measure of differential accessibility, and you compare to the actual FDR or the observed significance, you see that high predicted differential accessibility peaks in B cells are actually the ones that were computed if you have the data. And here is kind of a UMAP of showing one particular feature, how you're inferring. One example, how you are imputing the missing data. Another of the cool things was that, what is when you have a statistical model and you're trying to do classification, you care about, for example, calibration. And calibration means like how well you can, classify something given the uncertainty that you are giving on the result. If you are really certain, you don't want to make a mistake. If you are really uncertain, okay, like if I tell you, I'm not sure, okay, is the peak accessible? 0.5 plus or minus 10. Well, it goes from 0 to 1, so it's almost impossible, but you get the point. So calibration will be like a straight line here, which is meaning like you have certain uncertainty and certain error. You have low uncertainty and low error in this area, and for the ones that you have high uncertainty, you make high error. So if you don't tell much, I'm really uncertain, okay. But then there was this kind of tail that goes, which is telling you that the model is quite sure, and given the observed data that is hidden, because we hide the data and we predict it, like it's making a huge error. So why was that? We went back and looked, and there were some peaks that all the nearby cells were accessible, they were 1, but the cell was 0. It was sparsely like detected, and the model was telling us with high confidence, this is accessible. So it was kind of integrating what it should be doing, like and imputing from the nearby. All right. So from these two, and I have 10 more minutes, no? From these two experiments, I think that what I was trying to show you again is this organizing principle of the low dimensional being something that really encompasses the biology and is washing out the effects of modalities, and this high dimensional data and analysis being able to predict the missing data. Like, I was trying to illustrate again this overarching or like encompassing principle that I have shown. All right, so in the next, this is the moment I will need water, but in the next 10 minutes, I'm going to show you how we use this in a massive data set that we collected. Now, from there, I moved to the Allen Institute. Now it's the transition to really understand the progression of Alzheimer's. And I'm leading the analysis effort of what it's called the CAD dataset, the CAD Consortium, which is the Seattle Alzheimer's Disease Brain Cell Atlas, which bring together investigators from the Allen Institute, University of Washington, and Kaiser Permanente. And the aspiration of CAD, of this consortium, is trying to map the cellular and molecular changes that are happening as Alzheimer's disease progresses. Identify which one are the cellular types that are being affected or vulnerable to disease, and try to maybe map or reconstruct the progression of AD. To achieve this, we recruit a set of brain donors that when they pass away, they donate their brain, and we create a cohort of 84 donors from which we profile all like possible modalities or many modalities, like with Allen Institute kind of like precision. On the one hand, we profile measures of neuropathology or the pathology of Alzheimer's that is happening in the tissue in multiple regions in the brain. And then we take different nearby tissue slabs and we perform single-cell RNA-seq, ATAC, multi-omic, spatial transcriptomics. We did much more, but anyway, let's stay there. So the first paper that you could find in bioRxiv is pertaining to the analysis of one brain region, and it's called the middle temporal gyrus. So we decided, we have all this multimodal data, we don't know what to do with it, how to turn that data into knowledge into Alzheimer's. Let's focus on one brain region to really start developing the model. And I'm going to, I cannot pass this slide. Yeah, okay. Okay, and I'm just, we have an entire Nature paper with like almost, I would call 25 main figures. So for the sake of this talk and 5 minutes, I'm just going to tell you something about the analysis of multi-omic data at scale. Like we have millions of observations, like you can count like around 3 million cells on the analysis of these multimodal data. Again, bringing back this idea of studying disease and trying to identify what are the cell type perturbations that are going on as this Alzheimer's diseases progress. And to be able to do that, the first thing that you need is a reference framework. You need to know what are the cellular types in tissue, and then see how they change in disease. So to be able to achieve the cellular types, you can ask me more, but we needed two pieces. One was having a reference cellular taxonomy computed in neurotypical donors. So we have young donors, we compute a cellular taxonomy, we identify the cell types, and that's our reference taxonomy. But then sometimes this taxonomy is computed with means that it could be you gather all the data, you find differences in gene expression, you think this is a cell type. So we went one step further because of this requirement of being robust to disease changes, and we make what we call super types or robust types, which is we subsample the data in the reference and recompute with a classifier the cellular types that were in the original BICCN reference. And many of them, when you subsample, the data were not present, sorry. So that tells you that if you cannot find it in the reference donor, you probably will not see it across a cohort. These were just 5 donors of 84 donors across disease, etc. So those cell types, we prune them or join them with some other cells, like with nearby. So that's what Allen Institute had before this BICCN reference, which was really detailed at the neuronal level. But at the non-neuronal levels, in disease, I showed you at the beginning of the talk, the class, like there was a novel disease states that arise. So we need to enlarge the taxonomy, looking at them and identify disease-associated microglial state. Okay. Integrated, armed with this reference taxonomy that was computed purely on RNA, because that was the BICCN and that was all the knowledge we carry, we decided to move on to the multimodal integration. And here we were integrating around 3 million cells across all the technologies. And something of the power of this approach is that you have been analyzing single nucleus RNA-seq data sets for years. So you know how to QC the data, droplets, all the methods work well. In chromatin accessibility data, the story is not quite the same. So by having a latent representation that is joined, that allows you to visualize directions of variation, allow us to first do a detailed QC. So this was something quite obvious, but these were cells just originated from one donor. That donor was just attacked, and they were, poor quality. Okay, you are quite certain that all of these blocks you could remove them because you could interpret the variation in the data. Okay, so removing those cells and then integrating with MultiVI, we were able to annotate cells across technologies at multiple levels of resolution. So probably you are used to see cells at the level of, okay, this is a PB, SST cell, but at the really granular level of description of our taxonomies, an SST cell is composed of around 15 cellular types that were clearly identified, not only in transcriptional data sets, but also in chromatin accessibility, telling you that, this pattern for multiple modalities might be represented something actually like underlying true biological variability. We really then went back and look at multiple features like patch-seq experiments, which has morphology and electrical properties of the cell. And actually, many of these cellular types correspond to different SST types that have different arborization and connections, like double bouquet, etc. So all of these, start pulling information from multiple modalities allows you, we are seeing this more and more, to really capture the underlying biological variability in the data. Okay, and this will be the last slide, which is just capturing like it's not making justice to what we, but armed with all of these types, which are all of these columns, you can then compute the association of the type against cognitive status or measures of disease. Like ADNC is a measure of how advanced AD is in the brain. Controlling for sex, age, race, methods, because we put all the modalities here. And then it allows us to identify the cells. This is a minus size, so this negative effect size means that the cells are dying. The cells that are actually associated or dying as AD progress, associated with ADNC, for example. And even revealing the appearance of non-neuronal cells, like this novel microglia state. But now we have a really defined cellular taxonomy, so we can really identify and also provide a reference framework for the field and frame all these cellular changes that we are observing, throughout disease and throughout multiple cellular types. So, it's 7:59. So we are.
We have questions. I mean,
Okay, so let's leave it there. We have an entire paper, and this doesn't make justice. So let me use the last minute to that. We created statistical models to map the progression of disease. So what before was this association with ADNC, which is a variable that takes 4 values, now it's a continuous scale. By having a continuous covariate, we refine and we really enlarge the effect size, having more discovery power, validating these populations, but they even getting more. And because we are mapping disease progression, if you go back and check the paper, we have one last that I forgot to add, a figure in the paper in which we can time, given disease severity, when each of these populations are dying, and identify that the SSC cells are one of the earliest populations, together with oligodendrocytes, that are dying in disease. So anyway, I'm talking up in the air, but go and check the publication. So I think that this idea of having this framework of cellular types together with the detailed mapping of disease pathology really allow us to kind of reframe the field and time disease susceptibility. Okay, and with that, I want to thank all the people involved in the work. Most of them are all the CAD team, but it's also people from the Jordan group. Roman Lopez are mentioned a lot. He worked with a postdoc in the lab, Jeff, and Elliot is, he was there when I was there too. And many of the people, that help us, Tal and Rohan, work with me to develop MultiVI. Adam created STBI tools, basically. And, many of the people that were there. So.
Thank you. Thank you. Thank you so much. That was awesome. I think that was very fascinating to see the state of the art of single cell analysis and how we can actually leverage all of that data that we're generating in the lab. But so we're at time right now, but if maybe you can take 1 or 2 questions if you.
Yeah, I'm not sure if in the chat or you were.
Yeah, if there's any
There's one in the chat right now.
Yes, so there is a question. Is there any future plan for integrating metabolomics data for building the model?
Yes, but I moved there. I work in Alzheimer, and I'm rebuilding the team. Yes, but maybe the answer is who knows when.
You're doing PGMs and Bayesian networks. How far are you from being able to potentially do causal inference? Because you are looking for interventions.
Yeah,
right?
no, absolutely. Yeah. So Pierre has published and is doing a lot on that route. But again, yes, that's one of the directions we are trying to go. Also modeling disease, relationships between. We have this scale of disease progression, this timing, use it, to try to deconvolve like a causal effects. But one of the key things is that we don't have intervention. We can try to do interventions in an animal model, but all of these, this is the cool part, but the sad part too is that it's human tissue. Brain donors, you know, we want to honor them, use the best we can, but it's observational. So
You would have to do only do modeling.
yeah, and yeah, maybe infer, we can infer what is the best perturbation and then try to like, yeah, those type of average treatment effects, like of something like gene and then saying.
All right, well, thank you again. That was great. Yes, I think that's all for the questions, but